Hello, everyone. This is Tao Wu, Assistant Professor in Department of Molecular and Human Genetics at Baylor College of Medicine. Yeah, this is my great honor to share our discovery and stories with you guys. And I did my postdoc in Yale Stem Cell Center and Xiao's lab. That's where we make this discovery, find a totally novel DNA methylation in human and mouse. And before then, I got my PhD in Chinese chemical science. I trained in the genomics and the bioinformatics background. Okay, so now I will start to present my story. Here, you can see the title that tells you we found a novel DNA methylation, the N6 methyladenine in mouse and human. Okay, we are an epigenetics lab. So instead of show you the very old style Weddington diagram, I like this pinball diagram better because in this picture, it's exactly tell you the core ideas or concept in the epigenetic field is the dynamics. The DMTs, histone acetylase or deacetylase, or some unknown component here, all these epigenetic machines work together and finally make a decision and make the cell fate decision and go through the different development paths or reprogramming paths, and finally the cell into one state. And I feel why I joined this field and try to do the epigenetics research, because in this field, there's tons of the unknown or undiscovered component, and exciting field is left. So you can make your contribution in the future too. Okay, here what I show you is till today, what's the known DNA modification or so-called functional DNA modifications in mammalian genome. You can see here from beginning, we have the cytosine. That's the normal base in our body. And the 5-methylcytosine is the first one characterized in mammalian genome, the modification on the cytosine. That's a methylation on the carbon. And this one known for about over 40 years. And in 2009, Dr. Angela Rao just discovered 5-hydromysocytosine here. That's a oxidation and demethylation product from the 5-methylcytosine. And later on, Zhang and Chuanhe and Yang Shi, several groups worked together with them, and also Guoliang Xu's lab. Then they discovered 5-FOMU-C and 5-carboxyl-C, and finally, they discovered an enzyme called TAT1, TAT2, TAT3. They can demethylase the 5-methylcytosine step by step, one by one, and finally back to the cytosine. So it's a circle and finish this modification, demodification circle. And this already been done in the last 10 years, and the pathway enzyme is very clear. However, there is another question left in the field also for about 40 years. Even from beginning, they find the 5-methylcytosine. Someone asked, okay, do we have the methyladenine in mammalian system? Unfortunately, several groups try to searching for this for about 20 years, but before 2015, this N6 methyladenine is only reported in the E. coli, and no one discovered this even in eukaryotes. And in 2015, 3 papers published on Cell together from Chuanhe, Yang Shi, and Dahua Chen's group report this new adenine methylation in green algae, Drosophila, and cell elegans. That's the first time we find this methylation in the eukaryotes. And then 1 year later, we discovered this N6-methyladenine in mouse embryonic stem cells. That's we did this job in Yale Stem Cell Center. I'm the first author, and I did this job in Andrew Xiao's lab. So, okay, this slide seems very simple, but takes the whole field for about 40 years. And now the field is still moving on. And definitely for me, it's no doubt, we will find more and more undiscovered, unknown, novel modifications in our body. And hope you guys can join very soon. And okay, then I will tell you how we discover these novel modifications. When I did my postdoc in Andrew's lab, our first scientific project or question is try to figure out one type of the histone H2A variant, H2A.X. This variant is only contribute about 5 to 10% of the total whole nucleosome in the mouse embryonic stem cells. So that means they deposit to the genome, maybe it's by random, or they can target specific loci. So in my first published paper in Yale, we discovered, okay, the H3.X deposition pattern is really important for the Yamanaka 4-factor iPSL induced pluripotent stem cells to gain the fully pluripotency. That means this variant deposition is not random, and they have their specific loci they should target, and this target deposition pattern is important for the cell fate decision. And just to follow that work, we ask, okay, then what's the signal or mechanism on the genome will serve as a recruiting signal to recruit the H3.X to the specific or right loci on the genome? So, well, the hypothesis is very simple. For the H3.X deposition, either maybe you have some specific motif or sequence, or you have some maybe specific modifications like 5-methylcytidine. At that time, we know 5-methylcytidine, 5-hydroxymethylcytidine, 5-carboxy. So either way, maybe they can serve a recruiting signal to recruit H3.X deposit to specific loci, right? So the hypothesis, one word is special strings here for the special beads. So to test this hypothesis, the classical way is to do the H2A.X ChIP-seq, and if you doubt the 5-methylcytidine or 5-hydroxymethylcytidine is a signal, then you do the maybe whole genome bisulfite sequencing, then to correlate to these two and do the analyze to see they correlate each other or not. But instead of to do that, I try to do some killer experiment to answer this question directly. So that's why I use the Pacific Bioscience, the SMART sequencing, so-called single molecule real-time sequencing technology, coupled with the histone native chip. here, the slides just show you my experimental design. From beginning, we just do the native chip to pull down the H2A.X nucleosomes bonding DNA fragment with the H2A.X antibody. And different to the classical so-called Illumina second generation sequencing, you need to do the PCR, amplify your pull-down sequence, set up the library. Here, for the PacBio SmartSeq, we do not do the PCR, just add the circularized sequencing adapter to the pull down the native DNA fragments, and then load this on the smart sequence vial and put them on the sequence machine to start sequence. Here, I just explain a little bit of this sequencing technology. You can see here is so-called smart sequencing, the vial, and they call it so-called zero mode. I don't know what that means. And then you put your sequence, the fragment you want to sequence in the vial, and the PacBio reagents, the core thing here, they have a modified DNA polymerase. And when they add this polymerase, they bond to the sequencing adapters, then they can start to read your fragment sequence, tell you it's the ACGRT, the base information. Remember, at the same time, we didn't do the PCR. If you have any modification on these fragments, when the enzyme goes through the modification, it's well known the enzyme will take longer time to go through the modified A in compared to the unmodified A. So finally, this sequencer gives you two-dimensional information. One is the base information, TCGRA. Another one is so-called kinetics or dynamics of the enzyme goes through each base. So to calculate this so-called interpulse duration time of the enzyme goes through to read this base, you will know the base is modified or unmodified. So it's something like you drive a car from A to B, and then if it goes through directly or flat, it will be quick. But if you face some, the hill, then it will take a longer time to pass it. So, okay, then with this powerful single molecule sequencer, after our first round of sequencing, the shocking result come out. With help from the PacBio bioinfo team, they told us, sorry, we didn't see any 5-carboxy or 5-methyl-C or 5-hydromethyl-C, but instead we see tons of the modified A there. So at that time, no one knows we have the N6-methyladenine or other modified adenine in mouse or human genome. So from beginning, we thought, it's something contamination. But it took me about 3 or 4 months with different analysis protocol, and I figured out, oh no, this is something real. It's not some false positive or contamination there. At the same time, as I told you beginning, the 3-cell paper just published, and then suddenly we know, in at least eukaryotes, we have this modified A methylation. So, and then we just, thanks for the other 3 groups, and we know exactly, and the standard methodology in the field. So we just use the different approach, the UPLC mass spectrometry, and also methyl DNA IP sequencing, and different methodologies to cross confirm and to make sure our discovery is real positive. So here you can see we get the standardized, the N6 methylation A isotope label. And then we use this as a standard reference and characterize what we discovered in the embryonic stem cell. So you can see with our genomic DNA, it fits the internal standard, the N6 methyl A. So this just tell you what we see on the sequencer. It is the N6 methyl adenine. It's not the other modifications like 1-methyl A or something else. And from this quantification, you can see the H2A.X pull down, the IP samples, it is really enriched this methylation compared to the genomic background. So here, I just show you, instead of just discover a novel methylation, we also characterize the first demethylase of this N6 methyl adenine in mouse embryonic stem cell. That's so-called ALKBH1 gene. This gene encodes this ALKBH1 protein, and if you knock out this demethylase, you can see in the knockout cells compared to the wild-type cells, N6 methyl adenine level goes up. And here is the mass spectrometry quantification result. You can see after you knock out ALKBH1, either the genomic whole genome or the H3.X pull down part all has the more N6 methyl adenine methylation compared to the wild type condition. So it just means ALKBH1 encode the demethylase for this methylation. Yeah, from another angle, just to tell you, okay, this methylation is not something like ADAP or no useful. It just tell you they can be dynamic regulate, add on or delete, yeah, by the specific enzymes. Here, just to show you the whole experiment of the in vitro biochemistry assay to characterize the ALKBH1 as the demethylase of the N6 methyl adenine. You just synthesize the N6 methyl A oligos and then add the recombinant protein plus the cofactors together. And then you can see the ALKBH1 can demethylate this methylation. And if you add 1-methyl adenine, another methyl A, the ALKBH1 cannot demethylase this modification. So here is the mass spectrometry quantification for the demethylation efficient. So in vitro and in vivo both, we demonstrate ALKBH1 serve as a demethylase for this novel methylation. So after that, we just ask, okay, so what's the function of this novel methylation? Like 5-methyl-C, they can suppress gene expression. So how about this new methylation? So we did the RNA-seq, compare the ALKBH1 knockout and the wild type embryonic stem cells. Remember, ALKBH1 is a demethylase. So after you knock out ALKBH1, it means gain function of the N6 methyl A. So with RNA-seq, you can see it's very clear the 6-methyl A serve as a suppressor instead of the activator. The function reported in the other organism, like in the plant or Drosophila. So it's very interesting. Later on, we need to figure out, okay, why in the plant or Drosophila it serves as an activator, but in mouse or human it serves as a suppressor. Instead of the genes here, what I show you is I also analyze the so-called junking elements, or you call it junk elements in our genome. Is the transposons, especially the retrotransposons. Here you can see it's very clear for us N6-methyl A also not only suppress the genes, also for the retrotransposons, especially this line 1 family and the younger age line 1 subfamily elements here. And the line 1 contribute about 20% to mouse or human genome. So it's a superfamily of the retrotransposon. And the so-called young-aged subfamily of this retrotransposon, it just means they are still, they have the ability to jump in our genome. So it's very interesting to find this novel methylation specific target, these so-called active retrotransposon elements in the embryonic stem cells. So this slide I just to show you is one case with our DNA methyl DNA IP sequencing experiment, just to tell you, okay, where the methyl A locate in the mouse embryonic stem cell genome, and compared to wild type, you can see knockout, we have significantly increased signal. And here you can see this methyl A peak just sit down on the one younger line 1 subfamily, so-called L1MDA subfamily element here. And just note, we have another very important developmental functional element, DEX1, the gene DEX1. It's encode a very important developmental pathway genes. And so you can see these two is flanking to each other. So with several cases like this, it triggered me to propose a hypothesis model. So in summary, you can see what I show you. We discover a novel adenine methylation N6-methyl A in the mouse embryonic stem cell, and it's demethylates LPH1. And we also find this one can target either genes or mainly on the retrotransposon elements, and they can suppress the transcription here. And after you knock out LPH1, the demethylase N6-methyl A level goes up, and main part target to the young age family of the line 1 elements, then they can suppress their transcription, maybe jumping too. And also you have tons of the developmental functional genes flanking to these line elements. So the line elements to us is just to serve as some, the anchor element here. Then we hypothesize maybe methyl A here can reorganize the chromatin context, like maybe form heterochromatin, flanking this element, then to cause the flanking genes transcription shutdown. So that's the working model in our 2006 Nature paper. So, okay. After that, we just ask a naive question. So it seems in the normal tissue or body somatic cells, no 6-methyl A there. So we have some hints to say, okay, maybe in the ultra early embryonic development, this modification maybe serve as an important function there. But yeah, we don't have enough material to figure out that question or hypothesis yet. Instead of that, we just predict, okay, instead of the normal tissue or cells, maybe in the abnormal ones, like the cancers, could we find these modifications or not? Because remember, the epigenetics landscape is just like a hill. The normal development, the cells just go through and take several so-called normal somatic cell state. You can see the basin here just to show you the normal state. But remember, we also have some rare unnormal cell state. So that maybe show us it's the cancer cells or something weird cells else. And so in this state, you didn't find sigma-methyl A, but it's not means in those weird states, you cannot find them. So we collaborate with Dr. Jeremy Rich lab, which in UCSD now. Jeremy is the pioneer scientist who just defined the glioblastoma stem cells. And so we collaborate with Jeremy's lab and take advantage of their clear and well-defined glioblastoma stem cell system, and to characterize the sigma-methyl A function there. So from beginning, we get a very shocking result, and it's promising because you can see here with the dot plot in the glioblastoma stem cell lines, and even in the primary glioblastoma tissues, compared to our normal control human astrocytes, the sigma-methyl A level reach a very much higher abundance. And this is the mass spectrometry quantification. You can see, I didn't mark the mouse embryonic stem cell level here, but I can tell you the 1000 ppm here is around about 300, 500 fold higher than the sigma-methyl A level in the mouse embryonic stem cells. So it is ultra high. So it just triggers us, okay, to see where are they and what's the function there. So here I just show you this is the immunostaining with the antibody. And you can see they are indeed in the nucleus. This is after RNAs, RNA-T, RNA-H treatment. So very low or no RNA modification background. And you can see it's overlap with within the nucleus, perfect. And here is the IHC figure. You can see in the glioblastoma, the sigma-methyl A level is much higher than normal brain tissues. Here is the quantification of this figure. So it's in the disease tissue is significantly higher than the normal brain tissue of the sigma-methyl adenine. So then we did the whole genome methyl 6A IP sequencing, and then you can see sigma-methyl adenine is biased, heavily enriched, overlap with the heterochromatin part, which we mark here is the H3K9 trimethylation heterochromatin domain, or H3K7 trimethylation domain here. And in the heat map, you can see it's very clear sigma-methyl A and K9 and K7 is co-localized together. So that's just indicate it seems sigma-methyl adenine in the glioblastoma may be served together with the heterochromatin components or enzyme to hold or form the specific chromatin complex in this tumor cells. So we also check the LQPH1, human LQPH1, and we confirm it also serves as a sigma-methyl A demethylase. You can see we knock down the LQPH1, the sigma-methyl A increase, and also we did the RNA-seq. You can see after LQPH1 knockdown, sigma-methyl A level goes up in the glioblastoma, and you can see more downregulated genes. It's just to tell you, okay, it serves as a suppressor. in the glioblastoma. And here you can see after you knock down the demethylase, sigma-A increase, then you can see much more H3K9 trimethylation. We call it gained peak or increased domain compared to the decreased or lost domain. So it just means somehow sigma-A help or to collaborate with the H3K9 trimethylation enzyme to form the more heterochromatin domains in the glioblastoma cells. And this slide just to show you, okay, after you knock out LQH1, then you see tons of the increased 6-methyladenine peaks also co-localized with the heterochromatin body, the H3K9 trimethylation domain, and compared to the genomic background is heavily biased or enriched in the H3K9 trimethylation heterochromatin domain. And here, just to show you, somehow we can use the heterochromatin increase or formation to explain our downregulated genes. You can see about over 80% of the downregulated genes targeted by sigma-A is overlapped with H3K9 trimethylation heterochromatin domain. So it just means after you knock out the demethylase, sigma-A increase and help to form more heterochromatin, then downregulate those important genes correlate to the neurodevelopment or another very important function I will show you in a minute. So then with Jeremy's help, we just inject the LQH1 knockout cells into the mouse brain to test the tumor formation. Compared to the wild-type control, you can see it's some of the results from beginning we imagine it's opposite. So if you link sigma-A, higher sigma-A linked to high-grade glioblastoma, you will imagine, okay, after you knock out the demethylase, sigma-A increase should kill the mice even sooner than the wild type, right? But here, what I show you is after you knock out demethylase, then you inject the knockout cells into the mouse brain. They show you, okay, they rescue the mouse, and the sigma-A songs impair or compromise the glioblastoma stem cell tumor formation ability. Then we ask why. So I analyzed the LQH1 controlled genes with the gene oncology. You can see here very clear the LQH1 control tons of the hypoxia response genes in the glioblastoma cells. So it just tell you, okay, maybe we need to test the LQH1 target size and these genes, what the sigma-A looks like. And we also stress the glioblastoma cells with the hypoxia condition, 1% oxygen compared to 20%. So I show you one case here. It's very clear, tell you why this LQH1 knockout compromise the glioblastoma stem cells tumor formation ability. You can see XIAP is one important hypoxia response gene. So when you challenge or stress the GBM cells, stem cells, the XIAP should be upregulated, increase and express to respond this stress. So here in the normoxia, you can see sigma-A peak here. This is something like the silencer or suppressor, so they can suppress the genes. When you culture in the hypoxia condition, give the cell stress, then if it's wild-type cells, LQH1 can target here to demethylate this sigma-A and activate this gene's response to the hypoxia stress. But remember now in knockout cells, so you don't have this demethylase. So when you challenge or you inject the cells into the brain, it is always it's a hypoxia niche or context in the brain. So the tumor need to activate this gene, but they don't have this demethylase here can demethylate enzyme methyl A, so the cells cannot respond to the hypoxia condition or niche normally, so they will die. So that's why when you knock out this demethylase, the cells instead of to increase or help the tumor formation, the LP1 knockout cell, they cannot form the tumor like the wild type glioblastoma stem cells. Okay, that's the whole story of the enzyme methyl A in the glioblastoma stem cells. Now we are just moving on, try to test, okay, the methyl A function in the other tumor models. And we also have some other work is going on just to try to figure out the important function of this new methylation in the mouse early development stage. So here I just show you a slide I cite from Shanka's review just published last month. All the known 6-methyl A knowledge we show here, but I mean, some of them may be true or some of them may be not because the field is just a start and keep moving on. So we need some time to figure out, okay, what's the real enzyme and do we have more demethylase or erasers, and also do we have the readers or not? So yeah, if you have if you become interested about the field, you can go through and to read this very good review paper. And finally, I just cite Shanka's sentence, try to summarize the status of the field. Yeah, overall, we have more and more evidence for 6-methyl A in the mammalian genome, but we still need further studies to clarify the ambiguities or some part like the writer, the methyltransferase is too elusive now. And very important, we need to develop more and more and improve the methods and approach to detect and mapping this new modification so we can get more and more clear to see what's the real function of this novel methylation. So that's exactly, I set up my lab in Baylor Medicine, and we are developing novel methods, and we also searching and collaborate to set up a different new model system, try to characterize the function and the new pathways with this new methylation in human disease such as cancer. Okay, that's all. Finally, thanks to my PhD postdoc advisor, Dr. Andrew Xiao in Yale, and my colleagues, and also our collaborators, Impact Bio and Jeremy UCSD. Without their help, we cannot get this discovery. And thanks to my funding source, especially the CPRIT. CPRIT is fantastic. Without CPRIT money, I cannot set up my own lab in Baylor. So, okay, that's all. Yeah, then I will be glad to answer any questions you have. Thank you.